Good morning, good afternoon, everyone. Thank you all so much for joining. Uh, my name is Ben Van den Ries. I'm the CDO over at the Digital Medicine Society, and today we have a journal club on a new paper by authors from the Conexa Health team and others on regulatory pathways for qualification and acceptance of digital health technology-derived endpoints. And we have Jesse and Elena here from the Conexa team. Before we dive in, a couple of quick housekeeping notes that are on the next slide. So please note that today's session is recorded. To ask a question for discussion during the Q&A phase, please either raise your hand using the Zoom function for that, and somebody on our team will unmute you and ask you to ask your question live, or you can type your question in the chat box, whatever you prefer. Slides and recording will be available after today's session. So on that note, we're going to dive straight in. I'm going to ask Jessie to introduce herself briefly, followed by Elena, and then Elena will take over and dive straight into a brief presentation on the topic of the paper.
Hi, everyone. Thank you for joining. I'm Jessie Backer, VP of Clinical Development here at Conexa Health, and a longtime fan and collaborator at Dime.
Hi, I'm Elena Ismailova. I'm Chief Scientific Officer at Conexa Health. My background, I'm actually a bench scientist, and I used to do a lot of work on developing biomarker assays because before I have gone full-time digital. I'm one of the co-founders of Dime, and in my previous life, I used to be a drug developer for a period over 15 years. So let's dive into our paper today. So today we are talking about regulatory pathways for qualification and acceptance of DHT-derived endpoints. Before we speak about the details of this publication, I would like to tell you a short story of how the paper came together. It is a collaboration, as you can see from this slide, of the group of co-authors that represent key stakeholders of people who work in this ecosystem. We have people from the technology sector, people who work or used to work in the biopharmaceutical companies, people who represent public-private partnerships, and a former member of the FDA. The paper came together in response to frequently asked questions. What does it take to be accepted by the regulators? What is necessary? And we felt that if we write a publication in the open access journal, which we did, that it will add efficiency to DHT implementation process, and it will be open to the entire community. Just to note, the paper is focused on sensor-based digital health technologies, DHTs. All other versions of DHTs are out of scope, and the paper focuses on US regulations. The majority of the authors are in the US, and the space of regulatory science in this jurisdiction is very well developed. So let's get into the details. I will start by telling you that there are two major pathways which sponsors can use if they are thinking of using a DHT-enabled technology for data collection in their clinical trial. The first one is qualification pathway versus drug development tools. You can find a lot of information on the FDA website. In a nutshell, this pathway foresees development a tool with very broad applicability because it goes beyond specific drug development product and can be used by multiple parties. Often this pathway is done by pre-competitive collaborations. The other pathway is investigational drug, IND pathway. This one is usually undertaken by a single sponsor, and it's specific for a particular drug development product. One couple of slides later, Jesse will give you more details about these pathways and where they are different. The key part of the application, if somebody decides to use a DHT-derived measure for purposes of regulatory decision maker, is the evidentiary package. And this evidentiary package includes, as you might have figured out, verification and validation evidence, which is key, but also very important feature is a determination whether a DHT-derived measure is a biomarker or it's an electronic clinical outcome assessment, and later I will explain why. And last but not least, that there is a variety of choices of products from which to choose when it comes to collecting DHT-derived data. Of course, it can be an FDA-regulated medical device, but this not as is a requirement, and this is one of the most frequently asked questions. There are a number of products on the market that were designed specifically to collect data in clinical trials that don't have a designation of a medical device, but they can be used. And there is also a low-risk general wellness products, also known as fitness trackers. Now I'm going to pass baton to Jessie, and she will give us more details about the pathways.
Thank you, Elena. So as you just heard, the primary purpose of our paper was to lay out the differences and similarities between these two regulatory pathways, to arm a sponsor with the information that they need in order to consider these two options. We did not make a recommendation of a pathway because, as is pretty much always the case, the answer is it depends. And it really largely depends on the four attributes that I'm about to walk through. And so these are four areas in which the pathways are kind of diverge and become quite different from each other. The first is the way the actual application phases work. So on the right-hand side, the IND pathway, this is the one that is probably most familiar to people here today. Begins with an initial submission and then iterative interactions with the agency from that point onwards. In contrast, the DDT pathway is very structured. It has three distinct stages: the letter of intent, the qualification plan, and the qualification package. And each of those stages acts as a gating item, essentially. So after each, the FDA will respond with their comments and a determination as to whether the applicant can then move on to the next phase. The second area where the two pathways differ substantially is in confidentiality. So, of course, the details of an IND are confidential until the drug is approved. On the other hand, the DDT pathway submissions are open to the public throughout. So the FDA maintains a database where you can search not only for tools that have been successfully qualified, but also tools that, for whatever reason, have not progressed all the way through. So you can read the letter of intent, for example, and you can go on and read the FDA's response at each of those stages. So that's one area where there is a huge difference between the two pathways. Number three is the scope. And Elena just alluded to this earlier. So the IND pathway is, of course, specific to that particular drug. And the evidence supporting the choice of the endpoint, in some cases, could be reused for a different application by the same sponsor. But by and large, it's fairly limited. On the other hand, the whole point of the DDT pathway is to allow multiple sponsors to use a qualified endpoint, or I should say a tool, as long as the context of use is appropriate. And so in practice, what that means is that the context of use tends to be broader. The broader the context of use, the more evidence is required. And then finally, there are considerations of burden and risk. For the IND, the sponsor is obviously responsible for every aspect of the DHT implementation, whereas in their guidance, the FDA points out that the scope of a DDT submission in many cases will go beyond the resources and capabilities of a single entity. And therefore they recommend, it's not mandatory, but they recommend formation of a consortium or a pre-competitive collaboration of some kind. That obviously means that the work and the cost is shared across multiple entities, but it also means that each individual organization has less control over the submission itself and how it all plays out. But where these two pathways do not differ from each other is the expectations of the evidentiary package supporting the tool itself. So I'll hand back to Elena to go through that.
Hey, let's talk about the evidentiary package. I mentioned to you that I will explain the difference in the evidentiary package between biomarkers, digital biomarkers, and electronic outcome assessments, ECO. But before we jump into the differences that are highlighted in this slide, I would like to discuss briefly what is in common. Of course, the package should contain the description of hardware, firmware, software, and technical specifications of DHTs to be used. Verification testing is a must because hardware and firmware may have been created for completely different purposes than the use in a particular clinical study. Validation of the data processing algorithms should also be described, which can be done either in healthy participants or subjects with the disease. The evidentiary package should contain end-to-end data flow description, how the data is being collected, where it's stored, how it's being transmitted, and the archiving process. Operational considerations are very important. Specifically participant instructions and usability data management and statistical analysis plan, of course, have to be included, along with a control that ensures data privacy, security, and retention. An important feature is determining how data can be attributed to the study participant, and if possible, the context of data collection, including, in addition to the timestamp, the environment in which data was collected, clinic or home, and if possible, the activities that study participant was engaging in when data was collected. The other important consideration is raw or sample level data collection whenever possible. The data processing algorithms evolve over time. They become better and more efficient. And if the applicant has an opportunity to store the sample level data, these data can be reanalyzed at a later time if better algorithms become available. Now let's talk about differences in experiments and evidentiary package depending on the DHT-derived measure classification. If it's a biomarker, which you know from the FDA definition, is an indicator of normal pathological process or biological response to intervention, then the package should include the biological rationale why biomarker is required and why a particular measure is a good biomarker, along with unmet need indicating why new measure is being required. And also association between the biomarker and clinically relevant outcome. You may have noticed when I was speaking about commonalities, I didn't mention clinical validation. That's where the key difference between biomarkers and ECO lies. And association between biomarker and clinically relevant outcome for biomarkers constitutes clinical validation. Technical performance should be also included, but in the case of biomarkers, it's largely covered by verification and analytical validation experiments. In the case of echo, it's different. First of all, the echo instrument should cover features of disease that are meaningful to patients according to FDA guidance on patient-focused drug development. The ability of an instrument to capture clinical outcome is an equivalent of clinical validation, and the participants should also be able to understand instruction. The scoring method is a must because an instrument is a certain number, and this number should be interpretable, and the instrument should show sensitivity of change to be able to capture response or lack of it to particular therapeutic intervention is intended to measure. There are a number of DHTs that can fulfill both categories, and I will let Jesse to talk what option exists in this space. Please.
So the one of the really a secondary objective of our paper was to provide some information as to the medical device regulatory categories where a DHT might fall. And hopefully this is particularly helpful to individuals with a pharma background who may be less well-versed in the FDA medical device regulations. So on the left, we have sensor-based DHTs that are regulated as devices in their own right. Most of the time, they will be considered legally marketed medical devices, but in some cases, they may be considered investigational. And there are three ways that might be the case. One is if the DHT just simply hasn't yet navigated one of the medical device regulatory pathways successfully. The second is the DHT has successfully navigated one of those pathways and it's been put on the market, but it's been modified in some way, either the hardware, if that exists, or maybe a different algorithm, things like that. And the third way is if the device is on the market and it hasn't been modified, but it's used in a way that falls outside its labeling. So in any of those scenarios, depending on the objective of the study, the investigational device regulations might kick in. Over on the right-hand side, we have low-risk products that support a healthy lifestyle, and the FDA refers to these as general wellness products. They do acknowledge in their guidance that some of these products may be considered class 1 devices. That's the lowest risk category, and therefore they are under enforcement discretion by FDA. And if not, they are simply some other kind of a general wellness product. These are products that are marketed directly to consumers and are not considered, they don't make any medical device related claims. So they're fitness trackers and some mobile apps and things like that. But there's also a category that sits in between. And these are products that are developed specifically for research-related data capture. They do not have a medical device designation, but they're also not marketed to consumers for healthy lifestyle purposes. So they don't fit either of the other two categories. The easiest way to think of these products, I think, is they are the sensor-based version of an ePro platform. It's not a perfect analogy, but it's similar in that they collect research data, which then comes under regulatory scrutiny, but they're not regulated as a platform or a product in their own right. So within each of these categories, there may be combinations of hardware or software only. And I won't go through all of these, but I'll just put some examples, generic examples along the bottom. So this is really, in addition to a decision on which pathway to pursue, the purpose of this figure, which is a version of figure 1 in our paper, as well as the text that we devote to this topic, meant to guide sponsors as they are considering which DHT to select from what is often a very crowded group of products. So I'll hand back to Elena.
Okay, and I would like to conclude this introductory portion of the journal club with a quote from our paper that application of DHTs in drug development is a really interesting intersection of pharmaceutical, medical device, and regulatory science that hopefully is reflected by our authorship in the paper. And this intersection is governed by multiple regulations and guidance documents in the US, raising a number of questions around regulatory pathways, how to make sure that a selected DHT is fit for purpose, and how to select it from wide variety of regulated and non-regulated products for data collection, and how the evidentiary package should look like. I would like to encourage you to read our paper if you haven't read it. And in conclusion, I would like to thank the outstanding team of co-authors and brilliant scientists, and thank Odd, Stephen, Chris, Joe, and John for joining in our endeavor. And we sincerely hope that this paper will help scientists to design new experiments and leverage DHTs for the purposes of drug development. Thank you.
Thank you so much, Elena, Jesse. That was great as always. If I need to, well, nobody's asked me, but if I try to summarize the paper in one sentence, not to do it a disservice, but to really focus on at least what for me was the main highlight, is that what you tried to do was add efficiency to the process, encourage use, right? Lay it out in such a way that it encourages people to say like, okay, this helps me find my way and understanding what options are available to me when I'm considering adding digital health technology to a trial. We all know that there's many pilot studies and feasibility studies out there, but when it comes to clear examples where a DHT was used to measure a primary endpoint, then we definitely have only a very few examples, and clearly there's work to be done to make sure that people find their way in that direction and know which pathways are available to them. So I would ask the audience to please participate and add questions to the chat or raise your hand, and we will give you the mic while people think about what questions they may have. To kick things off, I believe it was you, Elena, or Jesse, it was you that was talking about the in the DDT pathway that there's a recommendation from the FDA to use a consortium or a pre-competitive collaborative group of companies and stakeholders, right? And that might be advantageous to tackling a DDT pathway. Can you tell or speak a little bit to what type of consortium you believe could see a DDT pathway through successfully, and why a consortium like that would consider taking that route?
Yeah, absolutely, Ben. So I mean, first of all, I'll just acknowledge that it is possible for a single entity to pursue the DDT pathway. Absolutely. And there's precedent for that. But getting back to the considerations of burden and risk, yes, forming some kind of, it might be a private consortium, a public-private partnership, a pre-competitive collaboration. So there are different combinations of bringing multiple organizations together. But the key is to have representation from multiple stakeholders, not only because of the ability to share cost, share experience, leverage different areas of expertise, but also, and I didn't get to this in the presentation, to support adoption. If a number of different organizations rally around one particular drug development tool, a biomarker or a COA, and make that evidence-driven decision, and then follow that pathway through to qualification, it means that adoption is more likely to occur and be widespread, as opposed to individual organizations developing biomarkers and COAs that are, measuring largely the same concept of interest, but are different from each other. Elena, anything to add on there?
You described
Okay.
perfectly.
And going down that path a little bit further, if you're talking about driving adoption, right? I fully follow that argument. And if you're like in your experience, based on the work you did for this paper, but also just your professional experience, what kind of stakeholders do you believe should be in such a consortium to really drive adoption, right? If you look at it from a multidisciplinary perspective, in your quote, Elena, at the end, you really highlighted that this is really a very interesting interface of all these various fields. And of course, one assumes that therefore a consortium like that should probably reflect that multidisciplinary aspect. But is that the case? Is that your thinking? Is that what you've seen happen?
I'm not aware of any specific examples that would fit exactly the membership that was on our the membership of authors on our paper. That would be ideal, in my opinion. The key driver for the process would be the technology providers and pharmaceutical companies that ultimately take full responsibility for what goes into their study protocols, but it's probably best done on the best of public-private partnerships that with early engagement from regulators and getting the feedback.
There's two questions in the chat. I'm going to jump to the second one first because it talks specifically about what we're talking about now, and then I'll go to the one above that. So Peter Fernandez is asking, don't see any single pharma taking the DDT route as it's very complex. The IND approach is straightforward, focused on DHT endpoint being targeted, and is confidential and exclusive. Your thoughts on how to get a consortium to collaborate?
Peter, that's a really good point. And indeed, the IND approach is more straightforward if the pharma sponsor has appropriate expertise and can form an appropriate good partnership with a technology provider. And I know where you're coming from because you've done it. And my personal admiration and applause to you and your team for doing such an amazing endeavor, because using MVPA for the purposes drug development and getting the FDA engagement is probably the most advanced case in the field, at least we know of in the public domain. And in my opinion, it's a huge win for the field. Thank you for creating and pushing the envelope. And this example shows, yes, under the IND pathway is totally doable. Where the DDT pathway comes into play is when the measure is complex. It will take years to develop and validate, and it requires multi-stakeholder experience. You are correct. I'm not aware of any examples where a single sponsor would do such an endeavor and get to the finish line. That's why it's probably done best in collaboration, public-private or private consortium.
All right, we have another question in the chat that's switching gears a little bit. Just to make sure that we get the question and the nuance of it right, Piper, do you mind coming off mute and giving voice to it live?
Hi, thank you very much for that presentation. In the bit where we talked about the different types of evidence we might want for ECOA versus digital biomarker, there was one part where, like, for ECOA, we want sensitivity to change and we want interpretation thresholds. And that's psychometrics. We do this whether it's a paper-based questionnaire, we do this in digital because it's all quality of life, for a digital biomarker, I can understand not wanting like patient relevant interpretation thresholds. We're dealing with a different thing, that's fine. Do we still, though, want to show evidence of sensitivity to change, or does it just depend on the type of biomarker you're interested in?
Thank you. This is a really thoughtful comment. As you know, there are many categories of biomarkers, and unlike COAs, they can be predictive, they can be monitoring, they can be efficacy biomarkers that makes them very similar to COAs and prognostic. And it really depends what the biomarker type is and what is the context of use for in drug development to go through the attribute. It's very much case dependent. We didn't go into that level of details, but I would strongly encourage you to go to the FDA website and look at the DDT qualification program. The information is, there's a lot of information there. As Jesse mentioned, it's fully transparent, and just get a sense of it, how the qualification package looks like, because it's all there, and what type of measures, biomarkers or COA have received regulatory qualification.
Yeah, thanks. I think it sounds like from what you just said there, it kind of just boils down to show your biomarker does what you think it does, right? Like that evidence for validity of the thing that you're trying to do. It's straightforward with ECOA approaches, but with biomarkers, it has to be a bit more of a horses for courses kind of approach. Thank you.
Yes.
Thank you, Piper. To switch gears a little bit, and this is probably one of those questions that usually is reserved for like closing things out, but I think it's important enough to move it up the chain a little bit. Elena, Jesse, from your experience, both working on this paper, but also more broadly, do you believe that the DDT and the IND pathway sufficiently cover the needs of the field? What do you believe, if anything, the field may need next?
That is a tricky question, Ben, but I'm glad you brought this up. I do think that both of these pathways, especially given the purpose of why DDT was developed and implemented, I think that they do cover the needs of the field. As Elena mentioned in her introduction, the regulatory science here within the US is reasonably advanced, and the guidance is detailed. And actually, if you scan through the reference list of our review, a huge number of those references are to FDA guidance documents and the FDA website. So I do think that these two pathways cover the needs of the field, and there's good guidance to support them. And honestly, what I think what we need is time. We haven't seen in the US a drug approved on the basis of a remote DHT-derived endpoint. We have seen, we referenced this in our paper, one drug approved for a DHT-derived endpoint collected in lab. But part of the reason that we haven't seen DDT qualification of a digital endpoint or approval of a drug based on a digital endpoint is simply time. Elena?
I would add one more word. It's time and perseverance, because drug development is a trial and error. There is no way around it. And in a nutshell, it's a definition of the experiment. And in another paper that was actually written by a DIME collaborative circle that also includes Jesse and Ben, we compare digital measures to lab assays trying to find commonalities and differences, trying to kind of to predict the future where the digital field is going, and also trying to reinvent the wheel because certain problems have been solved in the past. And that paper clearly shows that it's time and the cumulative number of examples of what worked and what didn't work, and people continue trying until we figure out. I moved into clinical development in early 2000, and the field was very much looking like the digital field looking today. Now a biomarker assay is not even being discussed, is often added to the clinical study protocol by default. And if we generate enough data for regulators to transact on, put, like Bellerophon did, a lot of information in the domain to inform other people and continue trying, we'll get there. I'm confident.
And actually, if I can just add one more comment there, we have seen successful qualification of a medical device development tool that is DHT-derived last year. Now, our review focuses on drug development, so we did not get into the MDDT pathway, but Apple did receive successful qualification of their measure of atrial fibrillation burden in 2023 as a secondary endpoint. Now, the pathways themselves, the drug development tool and the medical device development tool pathways are different. The stages are different, the processes are different, the centers of the FDA are different. So it's not, there isn't an easy overlap, but we have seen really great precedent for successful qualification. And I'm sure that, it won't be too far away that we see the same thing happen within drug development.
All right. I very much appreciate that perspective from both of you. Down the same path, at least to an extent, if you would be asked, and I'm sure maybe sometimes you are, by a developer of a DHT that is who is specifically looking to position their technology for use in a trial, what are tips that you can think of that you would give them? And tips sounds a little bit, fluffy and easy. You've made a lot of recommendations in the paper, but like really from a practical perspective, if you're thinking of what are the pitfalls that both of you have witnessed, what would be your advice?
Yeah, so I'm actually just going to flip back a couple of slides just as a quick reminder. Oops, here we go. To this slide, which is based on figure 1, just as a quick reminder. Our paper does list some of the pros and cons of selecting a regulated medical device DHT versus a non-regulated medical device, a non-regulated product DHT. But just to emphasize, there's no reason why a medical device can't meet all of the pros of a non-regulated product and vice versa. So you basically what I'm saying is you can get the best of both worlds. And I would be very pleased if quite soon as a field, we move away from using the regulatory status of the DHT as shorthand for suitability for data collection purposes, or like, a green light stamp of approval. It's been a long time. CTTI made that recommendation, I don't know, 7 or 8 years ago. DIME has reiterated, and of course the FDA guidance clearly states that the DHT itself does not need to be a medical device. Where it is important is when the investigational regulations kick in, as I mentioned. But to get back to your question, the 4 big pros of a medical device DHT, number one, is leveraging the existing evidence. So the developer has some degree of existing evidence that they used for their submission. It's probably not sufficient for the sponsor, but it's there as a starting point to build from. Number 2 is the general and special controls that the FDA mandates be applied for devices depending on which class or risk category they fall into. So that means you can be assured that there is a quality management system, that there's good manufacturing processes have been followed, et cetera. Number 3 is that generally speaking, the raw data, the sample level data is available. And lastly, that medical devices have version control and change management processes in place. But on the other hand, the pros of a non-regulated product, and I am generalizing, admittedly, they may be perceived as being more aesthetically pleasing, more user-friendly, and that's not trivial. If that leads to better retention and better usage, adherence, compliance, whatever label you want to use, then that's a big deal. Participants may be more familiar with those products. They may even own one themselves. And generally speaking, the cost is lower per product. But the point is that it is possible to create a product meeting all of those pros and none of the cons, regardless of the regulatory status. So I guess my tip would be to try to do that, to develop a product that meets all of those needs and develop an evidentiary package aligned to FDA guidance that the sponsor can use as a starting point for their own submission.
I really appreciate that viewpoint, Jesse. It's also something that matches my personal experience, where often there is indeed a immediate fallback to, if we're going to be using this technology, it first needs to get through, it needs to have its 510(k) or something along those lines, right? Now, that said, again, from my personal experience, that push is almost exclusively driven by the sponsor. At least in my experience. Does that match your experience? And if so, or if not, what can you think of that would help sponsors or other parties who may be pushing for this see that indeed there is no need for this, provided you have sufficient risk management and other controls in place?
Yeah, Ben, I have also in my experience, I see that coming more from sponsors. And I, by the way, have a medical device background. I don't have a pharma background. So I've kind of been on that side of the table, if you like. For a long time, it was a bit ambiguous because the FDA, prior to their DHD guidance, it wasn't kind of set out in stone, this question or their approach to that question. So I think it largely came about as, I guess, a somewhat easy way of getting a check mark. But unfortunately, it does take a little bit more digging. So it does require a sponsor who may not be so familiar with digital products, with medical devices, the nuances of V3+ related evidence, to become familiar with all of those aspects of evidence generation and the acceptance and qualification needs, but also to develop a really good partnership with DHT developers who do have that expertise. And that's where I think, kind of coming back to the previous questions around multi-stakeholder collaborations, becomes really important. But Elena has a very different perspective with her pharma background.
Yes, I will come from the perspective of my previous life in biopharma world. And I would say that for an extended period of time, the DHT-based devices and drug development were parallel, separate worlds. And somewhere probably in early to mid first decade of the 20th century, they started converging when people realized that there is efficiency on leveraging DHT's tool, and that started process of education on both sides, what is required for drug development clinical trials, and on the other hand, what is required from the device to match it. And of course, people who haven't lived through this experience looking for some way to get the regulatory endorsement, some kind of stamp of approval. And one of the goals of our papers was to educate and explain that just a stamp of approval of a medical device does not necessarily fulfill all sponsors' needs. It's more nuanced. And as the FDA representatives reiterated many times in public, the context of use drives everything, because it may be well working, cleared or approved medical device, but the context of use that sponsor is contemplating may be vastly different. And it doesn't mean that the information will be automatically absorbed into the drug development programs and the sponsor is all set. It may still require some work around that evidentiary package that we talked to a great extent, and our paper explains why.
We've been talking or approaching this primarily now from the DHT perspective. If you approach it from pharma slash sponsor perspective, where, because the field is changing and you often hear nowadays that there's even like more of almost a top-down push happening in some pharma companies to implement digitally derived endpoints into clinical trials, regardless of whether or not that's the right path to take, right? If you think about the recommendations that you would give to pharma and sponsors about implementing fit for purpose endpoints into their studies, that especially, well, not especially, exclusively in cases where the DHT version of such an endpoint really can come with efficiencies like you were describing previously. What would your recommendations be for teams that are looking to take that step but are feeling like it's like a big hurdle to take because there's so many unknowns? Not so much from the perspective of what do these technologies look like, what can they do, what's the safety profile, and so forth, but very much from the perspective of how can I design an endpoint that is going to be better because it's derived from such a technology as opposed to the default that I've been using for so long.
I would give looking at my old days in pharma when I was working on implementing DHTs, and it was much earlier in the field that we are today. I would give a brief advice to two things. Keep trying. Even if you have mandate from the top of your company, it's worth a try, because if you do the experiment, you will learn something, and it will be not limited to a particular clinical team. It will be an institutional learning, which is extremely valuable. And my second advice was look where is a good need for DHT measures. Not just don't be driven by a technology, be driven by a problem that this technology addresses, because it's key for drug development. And if you look for both PFDD guidance and biomarker qualification framework of the FDA, what is at the core of both these guidance documents is what problems do you address and how do you address it? That's what I would present that question. Be selective in what you undertake, not across the board, because resources are always limited, and keep trying.
Yeah, I mean, I as always completely agree with that perspective. And I'd also point out that it's developing a completely novel new measure that's captured by a DHT is very different to developing a DHT, I guess, version of a measure that already exists. In terms of the evidence required to bridge that gap, it's obviously much, much harder to develop a completely novel digitally derived measure of some kind.
Which is a very nice plug. Thank you, Jesse, for some other work that's happening at Dime on validating novel measures, and the fact that from an analytical validation perspective, that's a whole other ballpark in terms of demonstrating that, right? Okay, so I don't see any additional questions come in from the audience. If somebody still has a burning question, please come off mute and feel free to ask it now. In the meantime, thank you so much, Jesse, Elena, to kind of round this out on what next steps here are, or what next steps there could be. And could you please go to the next slide? So you laid out very clearly the DDT and the IND pathways. And I think we're all in agreement that although they are robust that, to your point, there's a lot of work to be done, and we have to stick to it to make sure that these type of technologies and this type of approach moves from where it is right now, which is kind of innovators and early adopters, to the early majority, right? To use that model where, you need to cross the chasm with any product. This isn't a single product. This is a whole field and a whole set of regulations behind it. But nevertheless, the same concept applies. We need to be able to move from early market to mainstream market and make sure that all the hard work that's been done by innovators and early adopters is picked up and used and translated in such a way that a much larger group of stakeholders are willing to take the plunge and willing to experiment with it, stick with it to demonstrate that it in fact can have a much larger impact on our entire field. And so I wanted to take a quick minute to call out that DIME has received funding from a broad agency announcement by the FDA for a project that started literally last month. So the work of this paper is incredibly timely and is absolutely the work that we're planning to do in that project is absolutely going to be an extension of it to build a how-to guide about the acceleration of implementation of DHTs in clinical trials, right? Both from the perspective of DHT developers as well as DHT adopters. What are the resources that currently exist from FDA guidance? And you referenced that in your paper, like the majority of references are based on FDA guidance and other FDA resources already. So what is out there already, both from FDA as well as other sources? And how do we put it together into an interactive process map that if somebody who's newer to this and who's trying to find their way either from I am developing technology and I need to implement it, or I want this technology in my trial, where do I start, right? Like, where do I go in the process map? And what you laid out in the paper with the DDT and the IND pathway is a great starting point for that. Where do I go? How do I find my bearings? What information, what resources are out there for me? And importantly, what the project is also going to do is identify where there may be gaps, right? Where maybe the resources that we have available are not fully developed yet, and we can really take that information, identify those gaps, see what we would need to fill those knowledge gaps, and again, make sure that as a field, we can accelerate the pace at which we're implementing these resources in a fit for purpose, these technologies in a fit for purpose ways into all of our trials. So wanted to quickly highlight that if you want to stay up to date on what's happening there, that will be posted on our social channels. You can find information on the website, and you can always reach out to me or other people in the team. And I believe somebody dropped a link in the chat too, direct to the webpage. Then final announcement to round us out on the next slide. We have a launch event coming up, a very exciting launch event on International Regulatory Pathways, or IRP, which will happen on November 19th. You can scan the QR code to get more information. Again, if you go to our website, you will find the same. You can register for the event. If you've this is going to be especially relevant to you if you've ever been involved in selection process of a digital health technology. You know that the applicable international law. So if you're going not just within the US or within a European country or even within the EU, that the applicable international law is really a patchwork of regulations, health technology assessments, clinical evidence requirements that are very diverse, let's say, and navigating that is not for the faint of heart. And the IRP project has built resources for a number of geographic areas to really lay this all out clearly, again, in kind of like a process map style for people to find their bearings, know where the right information is, know how they can leverage this, where the similarities are, where the dissimilarities are between the various regions, and how you can use that information to really set up a study in a digital health technology for success. So if you didn't already register, please do. The information is currently also in the chat channel right here in Zoom, in case you haven't registered yet and this is of interest to you. I didn't see any additional questions pop up. So I think we can I can round things out here by by thanking Jesse and Elena. I'm just getting, oh, I'm seeing the comment from Peter that he about the slide with the chasm. This is not an us thing, by the way. This is a model that's pretty widely used. The innovation diffusion curve that you can find to be used in quite some context, and I agree, Peter, that is very applicable in this context. So Jesse, Elena, thank you so much for all the hard work you put in this paper together with your team of co-authors, for walking us through what you've done here, for answering all the questions, including from the audience. I hope it was useful to you as well, and looking forward to seeing you at another DIME event.
Thanks, Ben. Thanks, everyone.
Thank you for inviting us.